Well, good afternoon, everyone. I'm Jeff Kelly, and I'm in the chemistry group here. And I'd like to give a slightly different talk today rather than talking a lot about my research. I'd like to actually advocate for an emerging group that we're creating that's focusing on neurodegenerative disease, new medicine initiative. And this is something that Stuart Lipton and I have been thinking about for quite a while, and he's the co-director. So I think it's fair to say that neurodegenerative diseases are a global health challenge. They're soon to eclipse cancer as the second leading cause of death behind neurodegenerative diseases. And that's partly due to the progress that we've made in treating cancer, but perhaps more dramatically due to the fact that we have an aging population who's more susceptible to these diseases. So we look at this initiative as being critically interdisciplinary, both in terms of the scientists involved and also the funding mechanism. So we're going to certainly be very aggressive to seek as much NIH money as we can, but that's not going to be enough. We definitely need philanthropic support, we need industry support, and biotech and pharma support as well if we're going to be successful. So I thought it might be useful to give just a little bit of my background for perspective. So it's taken us 25 years to develop tafamidis from the perspective of learning enough about the aggregation mechanism to be confident that slowing down aggregation was going to slow down the progression of the disease, and then coming up with a small molecule that stabilized the transthyretin protein. And unlike what Pete set up in Caliber, there was almost nobody interested in helping us convert our basic science information into a drug. And so the good news is I learned a lot along the way. The bad news is it took a long time. And it won't be until this year, hopefully, Pfizer will get approval for tafamidis for a couple of different indications, namely cardiomyopathy and peripheral and autonomic neuropathy. So I think what's most important is what have we learned, right? What we've learned is that the responder rate for tafamidis for polyneuropathy and autonomic neuropathy drops from about 75% to 45% if you're in the placebo group and then you roll over to treatment. So if you only delay the onset of treatment by 18 months, you lose about 30% of the population in terms of how much is going to respond. So treating neurodegenerative diseases early is absolutely critical. And I would argue that where we've really screwed up as a community is not going after early diagnostics in a huge way. And I think that's something that Stuart and I don't want to make a mistake the second time around. This is going to be a big initiative for us. And I envision this as being enabling for the whole industry. We look at diagnostics as sort of on the pre-competitive space, and our intention is to make what we learn available to everybody. Now, the other thing that I guess I would like to share with you is that transthyretin is one of 40 aggregation-prone proteins that lead to degenerative diseases. You could, in fact, probably discover a small molecule for stabilizer for most of these, but that would require a tremendous amount of effort. A lot of that's already going on in biotechnology and pharma. So what Stuart and I want to do with this center is to focus on neurodegenerative disease strategies that will apply to multiple neurodegenerative diseases. We're just not interested in a one-off strategy, given how difficult the clinical trials are for some of these disorders and so on. So wouldn't it be lovely if you had a strategy that would work for 5 or 6 major neurodegenerative diseases, and you could do a clinical trial in a disease where the endpoints are already well established, and then branch out, which I think is the strategy to use to go after Alzheimer's and Parkinson's, in my opinion. Okay. So to exemplify the kind of things that we're thinking about, we decided to apply to this Alzheimer's disease medicine initiative from NIH. This is something on the order of a $30 million initiative. And the whole concept was to first put together a really strong team. And some of the names that are on here you certainly recognize. Ian Wilson's a star in structural biology. Andrew Su is one of the few bioinformaticians out there that can handle omics information as well as pharmacology, which is really important, obviously, for these initiatives. Some of us have had the privilege of already developing drugs in the neurodegenerative disease space. And the whole idea is to enable the biotech and pharmaceutical industries and incentivize them to get involved in neurodegenerative disease again. That's the idea behind this program. And it's certainly our idea as well. Of course, also to enable Scripps, right? This is an initiative by NIH. I always forget what this is called. So it's the Accelerating Medicines Partnership for Alzheimer's Disease. They've spent an incredible amount of money coming up with 100 targets that are potentially useful for Alzheimer's disease, right? So the whole idea behind this grant is to come up with the technology that we believe exists within Scripps to ultimately validate these targets both through omics information, developing chemical probes for them, show that those probes change phenotypes in relevant models, and ultimately distribute the information in the probes to everybody to enable them to get into this space with targets that probably will ameliorate these neurodegenerative diseases. And I think this is a worthy project. It took us a lot of kind of months of thinking to put this together, but hopefully we'll get funded for this for NIH. But I want to emphasize again that that's not going to be nearly enough money to pull this off, right? But it's a good place to start. So to give you a sense of one project that might fall into this category, not making the same sort of lengthy drug development timeline that I had to follow for tafamidis, this time we collaborated with Caliper. Kristen Johnson and her colleague helped us develop what turned out to be a much more sensitive way to discover autophagy activators than has been pursued heretofore. Now, why autophagy? So autophagy is a process by which proteins, nucleic acids, lipids, things like the proteasome and organelles get degraded. And this ultimately occurs in the lysosome. It turns out that mutations in this pathway, virtually every step, cause a neurodegenerative disease. So the genetic basis for autophagy activation being useful in neurodegeneration is pretty straightforward, I would say. So through Caliper and their high-content imaging capacity, particularly Kristen Johnson, we've been able to pursue over 1 million small molecules using this high-content imaging approach. And we've now got about 50 molecules that activate autophagy through an mTOR-independent mechanism. And we think that's going to be really useful as a start to exemplify what we're trying to do with these other 100 targets that NIH has identified, one of which, of course, is autophagy. So I see that my time is up. I had a few more projects, but I don't feel bad about it. I hope I've shown you kind of the philosophy behind what we want to do and why we want to do it, and love to take your questions, I guess, after the whole thing is over. So, thank you.